start today's lecture. It's my favorite ever molecular biology subject. So if I do bad here, you have the right to boo me at the end, okay? Well, hopefully you like it as much as I do. So we're going to talk about epigenetics today. So one of the widely used example of epigenetics is in the course of the path to development and differentiation of cells. So what do I mean by that? So we know that the path to differentiation is stepwise and directional, and it involves changes in RNA and protein expression and results in a gradual loss of potency. What that means is that at one point in our lives, we were all just one cell, right? And then through all the divisions and development and differentiation, we became who we are. So that one cell is totipotent. What that means is that it has the potential to give rise to all kinds of cell types and tissues in our body. So as it divides in the embryo, after several divisions later, they become pluripotent. What that means is that they have the potential to give many different types of cells, but the number and the types of cells they can give rise to has been limited compared to totipotent. And at the end, what you're going to have is, at the end of all the paths to differentiation, you're going to have all kinds of different cell types and tissues that are differentiated. So how does this occur? It occurs by the action of certain transcription factors that control the path from, for example, totipotency to pluripotency to becoming a pigment or the nerve cells. So you have multiple of these transcription factors that control every step of this differentiation pathway. So what's striking about this is that our cells have exactly the same genome, but they have different structures and functions. So the differences between our cell types is not explained just by the DNA sequence differences, right? So, and what's more is that a liver cell, for example, once it became a liver cell, when it divides, it gives rise to more liver cells. It doesn't give rise to retinal cells, right? So there is a memory in the process that gives rise to these cells, that these cells remember who they are when they're dividing. So give me an example, and a disease example where the cells do not remain differentiated. That means they no longer remember who they are. Right? What kind of disease does this usually cause? Cancer, right? So
one example is neuroblastoma, where actually when the neurons actually resets to stem cells.
Exactly. So actually there are even grosser versions of this, which is teratomas. Not only the cell forgets who it is, it becomes pluripotent. So what you can have is you can have these tumors that look like portions of body parts, right? So you have these tumors on, if you type in on Google and say teratomas, you're going to see the grossest images that you're probably ever going to see, which is that the cancer cell forgets who it is, becomes more pluripotent, and differentiates into different tissues. It's gross. Anyway, so how do we define epigenetics? The epigenetics is, there is not a very good consensus on how we define this, but this is the best consensus that I could find looking at the recent reviews and interviews with people. And how we would define epigenetics is these are to explain the heritable changes that occur in the phenotype that does not involve changes in underlying DNA sequence. And the key here is heritable. When I say heritable, what I mean is it's heritable across cell division or to the new progeny. So this is where it's important to understand what heritable, how you define heritable is. All right, so what differentiates an epigenetic system from a genetic system is that epigenetic system is heritable, it's self-perpetuating, and it's reversible. So a lot of genetic changes are not reversible. Genetic changes occur through mutations, and if once that mutation is fixed in the population, you can't really go back, right? It's going to take a very unlikely event to fix the mutation back to its previous sequence. But epigenetic systems are reversible. So how do we know that actually, for example, in the case of differentiation, that differentiation can be reversed and reprogrammed in a cell? Does anyone know? How do we know experimentally that you can take a differentiated cell and reverse the epigenetic changes so it's no longer differentiated? Anyone heard of induced pluripotent stem cells? That's exactly how we know. So Yamanaka's lab in Kyoto, Japan, was working on this problem. So what they did is they were trying to see how they can undifferentiate fibroblast isolated from isolated fibroblasts. So their experimental system was pretty non-hypothesis driven. They said, okay, there's got to be something that we can do to reverse these back. And what they did is they expressed a set of different proteins in the fibroblast and then found that 4 proteins that when expressed in the fibroblast, that it confers these cells to be able to divide and follow a new path of differentiation upon proper signals. So what that means is you can take your fibroblast, you can transfect DNA to express a set of 4 proteins, and then it becomes more pluripotent with time. And then you can take those pluripotent or pluripotent cells and give certain signals, developmental signals, to push them to a new fate. Okay? So now this has really revolutionized a lot of the molecular biology field quite a lot. So what you can do is you can take, for example, these induced pluripotent stem cells and make neurons out of them, and people have been making different kinds of neurons and try to recreate some of the in vivo systems using in vitro cultured neurons. You can actually take these induced pluripotent cells and make a new mice. So not only they are pluripotent, you can make them totipotent, so that you could take those one cell and give rise to a mice. And not only you have given rise to a mouse, it can actually have babies. So you can create a whole new line of clonal mouse, right? Pretty cool. All right. Based on what we learned all through the semester, what kind of proteins do you think these 4 proteins that were expressed in the Yamanaka's experiment? Transcription factors. Those are the transcription factors are the key proteins that control cell fate, right? So it shouldn't surprise you that these factors were transcription factors. All right. So we said that the path to differentiation has some epigenetic systems, and there are some transcription factors that control these paths. And what was curious to people, and curious to a lot of people, is that some of these master regulators of cell fate, they actually get expressed during the developmental pathways, but then they stop being expressed in the differentiated cells, but the cells still maintain their identity. So what kind of molecular mechanism explains this? So this is the way we think about this. So for example, you have a gene that needs to be regulated in a certain differentiated cell, and that gene regulation is done through an epigenetic mark. We call these marks that can carry the information of a transcriptional memory, we call them epigenetic marks, and these could be certain histone modifications, not all of them, okay, and DNA methylation. So what happens is that at the beginning during the development, you have some transcription factors that are going to trigger the control of this gene. So they're going to bind to promoter or enhancer or both, etc., and they're going to set up the transcription of this gene, transcription status of that gene. So when these initial transcription factors are gone, the epigenetic marks that are put down on the genes are going to maintain the transcriptional status of that gene, even in the absence of the initial signal, which is the transcription factor itself. Okay? And these marks should propagate through cell division so that the transcriptional status and the marks will propagate through many cell divisions, and that's how the epigenetic information is passed from one cell to the other. All right. So as you can see, one of the key features of an epigenetic mark is self-propagation. That means after replication, it should be able to reinstate the mark again on the cell, right? Okay, so how does this happen? Let's take the example where we know most about this, which is the H3K9 trimethylation. We learned about this mark as part of the heterochromatin forming histone mark, right, that is bound by the heterochromatin protein 1, HP1. Okay, so let's imagine that you have a region of the genome. Let's say this is containing a gene that needs to be silenced, that has H3K9 methylation. And after replication, the histones that are deposited new, right, are going to be newly synthesized histones that do not have the mark. Okay, so all of these histone marks are actually put on the histones after the histones are deposited in the genome during the S phase. So the new histones are coming in. They do not have any marks. So what you're going to have is if you have a random distribution of the pre-existing histones in the two sister chromatids, what you're going to have is you're going to have genes with a region with less trimethylation. Now, this needs to be restored, right? And the way that happens is that the existing H3K9 methyl 3 from the old histones are capable of recruiting the H3K9 trimethylase, right? So this is through probably HP1, that is the HP1 binds to this methylation and brings H3K9 trimethylase. And once that trimethylase is recruited to these sites, it's going to methylate the neighboring histones in the neighboring nucleosomes, and now you have restored the methylation original status, even in the absence of the initial transcription factor that is triggered that methylation. Does this make sense? Okay. So what other property of heterochromatin actually depends on the ability of H3K9 trimethyl to bring more histone methyltransferase? This mechanism should not surprise you. You saw something similar to this before. Where did you guys see that? There was a key heterochromatin process that required the ability of H3K9 methylation, existing methylation, to bring more methyltransferase. I know some of you guys are really thinking hard. Think a little harder. How about spreading, right? How is spreading done? It was that through bringing HMT to spread more. Yeah? Oligomerization, not necessarily. The question that I'm asking is the ability to methylate a neighboring nucleosome, right, that doesn't have the methylation. That depends on the ability of existing methylation to bring more methylase to the region, right? And that occurs also in spreading. Does that make sense? Okay. All right, so let's get some concrete examples of this. And one of the examples were actually discovered in flies, and that is regulation of developmentally regulated genes through two protein complexes that are called the polycomb and trithorax complexes. And they got their names just like any other fly gene name by the phenotype that you see in the absence of these protein complexes. Okay, so these polycomb and trithorax complexes target hundreds of genes. So these genes control the structure and function of certain cell types in the body. Okay, so these hundreds of targets of genes contain a element, DNA sequence element that controls their transcription called the polycomb response element, the PRE. So first, during development, the initial transcription factors turn on or off these targets depending on what the developmental pathway requires. But then these transcription factors are gone, but the progeny of the cells continue to have the target gene on and off. And this is done through trithorax complex. For example, if the initial signal turns this gene on, it puts a histone mark called H3K4-methyl-3. This is a typo. It's not H4. It's H3K4-methyl-3. So the target gene is on. And if the initial signal turns off this gene, it recruits the polycomb complex, which puts the H3K27 methyl-3 mark, which is a heterochromatin mark. And once this gene is on, the maintenance of these histone marks that are active, that support active transcription, which is the H3K4 trimethylation, is ensured by the trithorax complex. So it maintains the active state of the open chromatin. And the repression of these genes is ensured by the polycomb complex, which keeps these genes in a heterochromatin state and maintains the repression of the genes through compact chromatin. Okay, so how does that heterochromatin repression actually occur? So this is a model by which we think the polycomb complexes work. So what you have is initially you have the gene that doesn't have any H3K27 methylation. So first the transcription factor, here is a repressor, recruits the polycomb complex, which contains this catalytic subunit, the EZ subunit. And that subunit of the complex methylates the H3K27. All right? So now once that methylation is put there, you no longer need the repressor because a second part of the polycomb complex called the PRC1 complex recognizes these existing methylations through subunits within itself, and once it binds there, it reinforces more methylation by putting in more methylation after cell division, as well as compacting this structure through oligomerization in a compact structure. So this PRC1 complex also has the H3K27 methyl-3 methylase subunit to maintain this methylation. All right, so if you actually took a bunch of nucleosomes on a DNA inside a tube, so this is an in vitro assay where you have you construct nucleosomes on a piece of DNA. Here you have 4 nucleosomes you can see on the DNA, and then you throw in the PRC1. You make sure that there is some methylation on these nucleosomes, and then throw in the PRC1 complex. What you can see is that these nucleosomes are now compacted, right? So this is basically a visual seeing is believing type of thing for this model. All right. Any questions about polycomb? All right. So how do we know that the polycomb and trithorax complex battle? This refers to my poor taste in movies. But basically, it's basically a battle during development on the regulation of individual genes. But how do we know that, right? And the reason that we know that is through analysis of mutants in trithorax and polycomb. So what we're looking here is the expression of the Hox gene called ABDB in Drosophila embryos. So this is one embryo in the wild type. The gene is expressed mostly towards the posterior of the embryo and not towards the anterior. So you can see the expression of the genes here, right? So if you have a polycomb mutation that makes a mutation in one of the subunits, critical subunits in the polycomb complex, what you see is that now the gene is expressed everywhere in the embryo, right? So the repression of this gene by the polycomb has been released, so it's relieved, so it's de-repressed. On the other hand, if you introduce a trithorax mutation, what you see is that the expression of the gene is now limited to a smaller place in the posterior of the embryo. So what's happening is that the polycomb and trithorax action is basically determining the battle towards with each other is determining where the repression is going to start, where the expression is going to start. It's determining the boundary between the expression and the repression domain. And this is really important because, as you know, the Hox gene expression determines where the organs are going to develop, right? So if you accidentally express a Hox gene, so these Hox genes encode for homeodomain transcription factors, what you're going to do is instead of making some wings in your thorax, you're going to start making wings in your head because you actually expressed a gene that controls, for example, wing formation, right? So that boundary of where things are going to be expressed is really important. We do not want a couple of arms coming out of here and a couple of arms coming out of our hips, right? So it's very important to control that. All right. So what happens when things go wrong with trithorax and polycomb? I just said that it causes a lot of developmental defects. Such defects in flies, you can actually observe them, but a lot of these very drastic defects in humans are usually lethal during embryogenesis, so it causes spontaneous miscarriage. But there are some also pretty interesting mutations like gives rise to 6 fingers, etc., and things like that. These are similar mutations. But we also know that the maintenance of gene expression is a part of the epigenetic mechanisms that keeps our differentiated cells differentiated. And if those mechanisms are affected, that can cause cancer. So one of the famous case of cancers is due to this gene called MLL, which is a subunit of the trithorax complex that is normally recruited to a set of target genes and methylates H3K4. But in a lot of cancers, this MLL gets fused to other proteins through chromosomal translocations. And once it's fused to other proteins, it misregulates its own targets and it ectopically regulates other targets. That means improperly regulates other targets, and that can cause cancers, which is, for example, one of the causes of mixed lineage leukemia, and that's how that gene got its name. Similar for polycomb. A lot of cancer cells have problems in repressing genes because they have defects in the polycomb complex. Okay, so these polycomb and trithorax complexes also occur in humans. So don't get the idea that it's just a fly thing. These are very well conserved mechanisms that occur across animals. All right. So that was one developmental example. And there are other examples of epigenetic processes. And one of the classical examples is a phenomenon that was originally named as true heterochromatin. It's called position effect variegation. Okay, so this was first observed in yeast and also in flies. So what's happening here is that in yeast, in a wild-type yeast, you have a gene called AD2, and when it's expressed and it's at its normal location on a chromosome, when this gene is expressed, the colonies that you get out of these cells on a plate appear white, right? So you probably saw some white yeast colonies. Now, there is if there is a null mutation of this gene in its normal locus, then that prevents its expression, or it causes the protein to be completely null, a mutant, right, non-functional. Then what you see is, due to not expressing AD2, these cells turn red through some metabolic pathway that I don't know, but they appear in red color. Okay. So what researchers have found was that they found a mutant where the AD2 gene was now moved near the telomere, right? The AD2 gene itself is not mutated. Only its location has changed. So from the center of the chromosome, it moved to the close to the telomere. So nothing is wrong with the protein or anything, right? But then what they realized is that now this genotype is giving rise to yeast colonies that have white sectors. So this is a real picture of this. What you can see is that it's white here, there's some red here, etc., etc. And the reason why these guys have sectors is that each yeast colony is started by one yeast cell. So you have one yeast cell, right? And it divides and divides and divides and divides. And each time it divides from the center of the colony, you're going to have a red, like a round-looking colony, right? Because everything is dividing. So let's imagine now, during that division, you divided, and that guy divided, and the other guy divided, etc., etc. One of these cells stopped expressing AD2, and it turned red, right? So if that cell turned back to expressing AD2, its progeny would be white, right? But what if that happens? That repression of AD2, if it's heritable through cell division, the progeny of that cell is going to be also red, correct? And the progeny of this cell, if it's heritable, is going to be red. And the progeny of that cell is going to be red and red and red and red, right? And that side and the other guys are going to be expressing AD2, and their progeny is going to be expressing AD2, etc. And what at the end is you're going to see a colony with a red sector, right? So that's how this phenotype is given rise. All right. So what that tells you is this. These cells have the same genotype, which is the same thing. There's no problem with the AD2 gene. But once the AD2 gene is turned on, that transcriptional status is inherited by the progeny of the mother cell, right? And if it's happened to be turned off, then the progeny of that mother cell inherits that transcriptional memory, right? So that's how that occurs. So that's a classical epigenetic phenomenon. We know how this kind of occurs because we know what might actually repress AD2 near telomeres. So tell me, we went through this in one of our lectures. We talked about the proteins that are responsible for heterochromatin silencing near telomeres in yeast. What are these proteins? It was in last year's midterm. So if you took the test midterm, you should remember this.
The SIR complex?
Yes, excellent. The SIR complex, right? So these proteins are the proteins that heterochromatize and silence AD2, and their action on AD2 is inherited to the next generation. Okay? All right, this also occurs in flies, and the phenotype that gives rise in flies is patched eyes. So in this case, what you have is you have a gene called white. So when mutated, gives white eyes, right? So in its normal location, this gene is expressed, and you normally have wild-type red-eyed fruit flies. But in a genotype where this gene is now located near a heterochromatin site, right, what you're going to have is some cells are going to repress this gene, and some cells are going to not repress this gene, and you're going to have this patch phenotype as here. Okay, here's a question for you. Why do some cells, what could be the mechanism of white gene repression when it's near heterochromatin? What property of heterochromatin might lead to white gene being repressed? We mentioned this just like 10 minutes ago. There's a property, spreading, excellent. If in some cells the heterochromatin spreads far enough to repress this gene, it's going to be repressed. In some other cells, the spreading happens to be not far enough, then that gene is going to be expressed, right? Okay. So that's position of effect variegation in flies. All right. So next what we're going to talk about is a classic epigenetic mark that is not histone modification. It's actually the modification of the DNA itself. We learned about this modification in the DNA repair lecture, which is that in animals like humans, what we have is we have most of our CPG sequences that C next to a G, the cytosine gets methylated, right? So that's cytosine methylation. So what you have here is that you have the cytosine and there is a methyl transferase that puts the methylation on the cytosine, and so this is a DNA modification. So in mammals, between 60 to 90% of all CPGs, so next to C next to G, are methylated. Except for CPG islands that are upstream of genes that tend to be hypomethylated. Hypo means not methylated that much. Hypermethylated means methylated a lot, right? And they're involved in transcription. We learned about these, which is the CPG-containing promoters, right? CPG island-containing promoters. So, and what we know in general, this is a general rule, but there are some exceptions involved, that in genes that contain CPG islands, heavy methylation of the Cs in that CPG island is associated with long-term silencing of that gene. Okay? So, again, this methylation should be inherited through a cell cycle, right? And you know that when the DNA is replicated, the new DNA strand is not going to contain the methylation. The nucleotides are not coming in already methylated. They are being methylated after replication. So what you have here is that you have a CPG where you have a C next to G, and on the other strand you have the same. So the methylation is usually symmetric. That means both C's in this CPG region is methylated. And when this parental chromosome goes through DNA replication and gives rise to two sister chromosomes, what you're going to have is one of the strands is going to inherit is going to be the old strand, right? So that's going to inherit the methylated C, and the new C that's coming in is not going to be methylated. So this is after replication. So we have DNA methyltransferases that maintain this methylation, and the way they do that is they specifically bind and recognize this hemimethylated region, which is this. It has only one methylation in one of the strands, and their binding now allows them to methylate the other C on the other strand. Now what happens is that after replication, the methylation status of this site is maintained, right? And so methylation status is propagated. So we typically classify the methyltransferases into two groups. It's a somewhat arbitrary classification, but it helps you think about it, the process mechanism a little bit. One is called the de novo and DNMTs. So these are DNA methyltransferases that are recruited to certain sites in the genome, and they put the initial methyl group without the requirement for a presence of a previous methyl group in the cytosine in the other strand. Okay, and then maintenance DNMTs basically copy the existing methylation in the old strand to the new strand. Okay, any questions about this? So in this example, what kind of DNMT is DNMT1? Is it a de novo or a maintenance? Maintenance, yep. So I'll also, sorry. I encourage you to go home and draw what would happen if the DNMT1 did not restore the methylation. What would happen to the methylation status of these two chromosomes after they go through a second set of replication, okay? And you'll see how the marks will be completely lost after a few divisions. So make sure to draw that out. All right. It's good to be the king. And so I have several highlights in the subject. One of the highlights is actually how to show you how DNA methylation can control the reproductive status of a whole organism. So epigenetic processes are not just in mammals and etc. and humans. They are seen in a whole host of different organisms, and in this case, the honeybees. So what you have in honeybees is that the honeybees that become in a colony, only one becomes a queen, right? Actually, there is no difference in terms of their genetic information between queens and the workers. But the difference is that the queens are raised in a specially constructed cell called the queen cup, which are filled with some kind of food for it called the royal jelly. Okay, sounds really good. So the larvae that develop into workers and the queens have the same genomes, but the queens eat royal jelly. And the royal jelly has enough, the ingredients of the royal jelly has that changes the methylation pattern of a set of specific target genes. So that methylation is done through the methyltransferase called DNMT3. So if DNMT3 is there, you methylate all of those genes. But if you make a honeybee that doesn't have DNMT3, and then let this honeybee eat lots of royal jelly, the bees grow up to be queen-like, right? They think they're the queens. So you bypass the methylation you bypass that process by having a DNMT3 mutation. This also happens in mammals. So here's an example for two mice that are genetically identical. So these are clonal mice. So their mothers were, while they were pregnant, the mothers were fed different diets. So the mother of this mouse was given a normal mouse diet. Apparently it's not very good, right? And the mother of this mouse was received a diet supplemented with choline, folic acid, and other things. So what happened is at the end, when you look at the DNA methylation amount in this mouse, you see that there's high DNA methylation. And this, when you look at the DNA methylation amount in this mouse, you see low DNA methylation. And this high DNA methylation is correlated with being more active and skinny. And not having enough DNA methylation causes obesity in mice. Okay. So that also, you can see that the coat color is different, right? That doesn't mean, again, correlation is not causation. This doesn't say the yellow coat color gene is making this mouse fat. It just says that there are other genes that control how fat this mouse is going to get, and they happen to also control the coat color gene called agouti. All right. So that adds to the mother's guilt even more, like they need. All right, so how do you actually map methylation in the genome? There are several techniques, one of which is asked in your study question, but I'm going to show you this technique. Yeah? Oh, okay. All right, so what you have is you have where you can map where in the genome the DNA is methylated. And the way that you can do that is you can take your cells, you can isolate the DNA, okay, and you take that isolated DNA and sonicate it into different pieces, smaller pieces. Then you can denature these DNA strands so you have single-stranded DNA, and you can use an antibody that specifically binds to DNA, but only when the cytosines are methylated. Okay? So you can make an antibody that specifically binds to cytosine methylated DNA, and when you do that, you're going to immunoprecipitate the DNA that contains the methylation. So this, after immunoprecipitation, you have DNA that contains the DNA corresponds to the genomic sites that are methylated, and if you compare it to the reference DNA or input DNA, where you do not do any immunoprecipitation, you can see what parts of the genome contains more DNA methylation compared to control, right? So what technique does this resemble? ChIP-seq, correct? So if you then take these DNA sequences and construct an Illumina sequencing library and sequence both the immunoprecipitated DNA and the input DNA, what you can do is you can go back, map the reads to your genome, and see where the coverage is higher and say, okay, that region of the genome is methylated more compared to the other regions. Any question about this? Okay. So another example of an epigenetic process that is developmentally essential is called the X chromosome dosage compensation. Okay. So X chromosome dosage compensation equalizes the overall X chromosome transcript levels between the two sexes. Okay, so in a lot of animals, sex is determined by the number of sex chromosomes, right? All the men in this room contain a Y chromosome and an X, so become XY, and the females have a 2X chromosome, that's 2X, right? So that creates a dosage imbalance. What that means is by just the fact that it has 2X chromosome in females, for any gene that's on the X chromosome, you're going to have twice the transcript amounts in females compared to males. Does that make sense? And that's actually not desirable because a lot of these genes on the X chromosome are equally important for both sexes, and they should be expressed at similar levels. Okay? So that creates a dosage imbalance for X chromosomal genes between sexes. Now, if that dosage imbalance is uncorrected, those embryos do not survive. So this needs to be corrected. And the way that it is corrected is through a process called X inactivation. Okay? So what happens in mammals is that one of the X chromosomes get almost completely silenced in terms of transcription. You basically inactivate one of the X chromosomes so that overall X chromosomal transcripts in the 2X females is now similar to XY males. In flies, the females do the work, the males do the work, which is very unusual, right? So what happens is that through the dosage compensation is done by males. So instead of inactivating the 1X in females, now the males activate 1X chromosome by about twofold. So what they do is they increase transcription of every X chromosomal gene by about twofold. And that equalizes the overall X chromosome transcript levels between males and females. Worms do it in a yet different way. So what they do is that in the females, which have 2X chromosome, both X chromosome transcription is downregulated by a factor of 2. So you basically halve the amount of transcription coming out of the 2X chromosomes. So at the end, you have the overall X chromosome transcription similar between the two sexes, right? So these dosage compensation mechanisms are, the strategies are different, but what we found at the molecular level, the mechanisms are very similar, and they all come down to regulating the chromatin structure of the X chromosome. Okay, and so you probably have seen X chromosome inactivation before in POB, etc., and you know that it is random. What that means is that so X chromosome inactivation in mammals occurs sometimes early in embryogenesis when you have several hundred cells, okay? And each cells make an autonomous decision, it makes a decision for itself, and it inactivates either maternal or paternal X chromosome. So if the cell inactivates this or that. So imagine that this cell has inactivated the paternal chromosome. So it's going to express only the genes that are coming from the maternal X chromosome. And this is an epigenetic process. What that means is once one of the chromosomes is inactivated, all the progeny of that cell, when it divides, it's going to keep that paternal X chromosome inactivated. Okay? So all the progeny of this cell is going to have the same X chromosome expressing. All the progeny itself is going to have the X chromosome, whatever choice it made is going to be expressing, and this one is going to express something else. And each of these cells are going to divide and give rise to different tissues, right? So some of these cells are going to be part of your liver, some of these cells are going to be part of your head, and whatever, etc. What that means is that the females that have two X chromosomes, right, we are called mosaics. What that means is different populations of cells in our body preferentially expresses either the genes from our mother or from our father, right? And the genes that might be expressed in our brain cells, population of brain cells, might not be the same as the genes that are expressed in our liver cells. So the males, you don't have that. The males don't have that. All right. One phenotypic product of the random X inactivation is the coat color, and that was how actually this process has been studied and found out that the inactivation process was random. So this is a picture of my cat. Sleeps a lot. Bothers me at 5 a.m. in the morning. But so what you can see is that this is a calico cat. And the reason why it's a calico cat is that some of the cells express the allele of one of the X chromosomes that happen to be brown, and some of the cells express an allele that happen to be black. Okay, so what that means in this patch of cells have arisen from one cell during embryogenesis that happen to inactivate the X chromosome containing the black, the gene expressing the black, the gene that controls the black color, so these guys turn brown, and etc. So what about this white, right? So that's interesting. So where's that white coming from? It's actually an unrelated autosomal gene that controls the migration of the melanocytes. So you have an autosomal piebalding gene that affects the migration of the melanocytes, and the expression level of that gene between different cells is actually different. It varies, and if the gene is expressed at different levels, if the melanocytes during development migrate slower, then you see wider fur, and if there's no whites and the melanocytes all migrated out, then you have a color, right? And if this autosomal piebalding gene is never expressed, then what you can see is all the melanocytes make it out, and then you see these tortoise kind of coat color. Does that make sense?
If the X inactivation occurred earlier in development, would it be like more uniform in color?
Exactly. Yeah. Imagine that the X inactivation occurred in one cell. Then everybody is the same, right? And it's very interesting. X inactivation is actually cannot occur in one cell. If you force it, what happens is that the cells lose their pluripotency. So that X inactivation process is really engaged to the timing of cell divisions. That means if you when the cells decide to come out of totipotency from pluripotency, they start the X inactivation process. And X inactivation actually is not immediate. That is gradual with time, and it takes a while to set it up, right? So it does not occur in very early embryogenesis.
Why is different dosages so bad? Like what?
Excellent. So gene dosage is really important because the stoichiometry of the proteins in the cells determines how well the cells can work. Imagine that you have a protein complex that only works. Let's imagine the simplest case where you have a protein complex with two subunits, okay? And imagine one of them is on the X chromosome. And let's say you improperly made one extra copy. But that extra copy cannot work, right? And that extra copy can cause several problems. At the very least, it could be a burden for the cell. That is, you basically produce something you do not need it, right? And what happens is that proteasome pathways actually degrade that protein. That's pretty well established now. So it puts burden on the proteasome pathway in the cells. It could have also dominant negative effects if it actually interacts with something else in extra amount, right? So the dosage of every protein in the cell is actually quite important. Now, the dosage of single genes are less important than dosage of complete chromosomes. That is, the cells can tolerate having an extra one or two protein around. But if you now have thousands of genes, and in humans, I can't remember, about 1,000 genes on X chromosome, now you have thousands of proteins that are produced extra because you didn't inactivate the X. Now you have a problem, right?
There's no problem with the fact that men have extra Y, like that doesn't cause also dosage problem.
Y is very gene poor. You guys really don't have much on the Y. Right? That's why actually a lot of X-linked genes that cause disease affect men, right? They don't affect women. And because you have only X chromosome, and if you happen to get stuck with the wrong allele, that's it. And you don't have much on the Y that's going on, except a few sex determination genes to make sure that the man have grow up and grow the right gonads and stuff like that. Anything else? All right, cool. Can you predict the coat pattern of this cat's identical twin if there was a clone of this cat? No. And the reason for that is completely random, and it's in later embryogenesis. All right. How can you look at a phenotype in a mouse? For example, you want to look at the mosaicism in the mouse, and what you can do is, what has been done is, people have created a mouse line where they have integrated two reporter genes, a red fluorescent protein gene and a green fluorescent protein gene in the maternal and paternal X chromosomes. And since one gets inactivated and the other one gets inactivated, each cell in one of these tissues is going to express either the green or the red. And you can get these beautiful pictures out of the mouse showing what genes, what cells are expressing the maternal or the paternal allele, right? So cool. All right, so how does that X inactivation happen molecularly? It happens through the expression of a long non-coding RNA called exist. Okay, so this is the active X chromosome, XA, and an inactive X chromosome. So you have two chromosomes in the females, and this is the X inactivation locus. It's a simplified version of that. It encodes for two long non-coding RNAs. One is called exist, and the expression of the exist RNA starts the inactivation process in the inactive X chromosome. And if the T6, which is exactly the opposite of exist, right, which is the antisense non-coding RNA that goes into the exist gene, is expressed, then the exist gene is repressed, and you do not make that long non-coding RNA, and that X chromosome stays active. So if you do an RNA fish, using a red probe against the exist RNA, what you can see is that you see only one spot in an XX female cell because only one of the X is inactivated. And then if you use a DNA fish probe that is going to hybridize and look green in is going to recognize the two X chromosome. You can see that one X chromosome is here, the other X chromosome here, and then you overlay the two pictures, you can see the X chromosome that's inactivated is here, and the active X chromosome is over here, right? All right, how does this occur? It's a gradual process. It starts with the expression of the exist from the X inactivation locus on the X chromosome. And once the exist is expressed and this wiggly red non-coding RNA, it starts spreading along the X chromosome. So that spreading is thought to be mediated by the three-dimensional conformation of the X. That is, the first region that the exist spreads is the closest site in the 3D to the X inactivation locus, right? So it's going to spread out here. And then once it spreads through, somewhat in the middle of the process, what you see is that then the exist spreads even more, and it coats the entire X chromosome. Now, how does a long non-coding RNA actually repress transcription, right? And that's a question that's been kind of probed and answered in the past decade or so. What turns out is that the exist RNA forms a pretty spectacular secondary structure that serves as a binding site for a whole bunch of heterochromatin proteins. So these heterochromatin proteins bind to the exist RNA, and because exist RNA is coating the X chromosome, they get recruited to the X chromosome. And once they're recruited, they do a whole bunch of things. One of these things is the trimethylation of lysine 27, which mediates polycomb-mediated repression, right? So polycomb-mediated repression is acting in X inactivation. So that represses genes. And also the recruitment of a histone variant called microH2A, which also represses transcription, and DNA methylation of the genes, which you just learned is associated with transcriptional repression as well. So it looks like the exist is the landing pad for any mechanism that represses transcription. It's going to bring it to X, make sure that the X is silent, completely silent. Okay? Does that make sense? Any questions about this? All right. Here is a question. What's a non-coding RNA? Give me an example of a non-coding RNA that acts as a structural component of a protein complex.
RNA,
Ribosomal RNA, right? So the exist activity is not unprecedented. There are examples of long non-coding RNAs acting this way. And it turns out exist is not the only one. In the past 5 years or so, researchers have also found other long non-coding RNAs that are capable of recruiting transcriptional regulatory complexes through their secondary structure. All right. So what? Can we actually apply this to anything? Actually, it turns out you can. So this was one of these high-risk, high-reward type of projects that the National Institute of Health granted and supported by giving money to the group to do it. So what they did is that they said, can we apply X inactivation to the Down syndrome? Okay, you guys know that the Down syndrome is caused by having 3 copies of chromosome 21, right? And we just learned that the gene dosage can cause problems. And the Down syndrome is caused by the improper higher expression of the genes on chromosome 21 compared to all the other chromosomes which have only 2 copies, right? And that's the reason why the Down syndrome is seen. This kind of chromosomal abnormalities actually is pretty common in humans. Humans are terrible in meiosis. I don't know how as a species we actually survive. that a lot of the embryos that are made contain wrong numbers of chromosomes. But because chromosome dosage is very critical for development, a lot of these embryos get spontaneously miscarried. Okay? Only a couple of them actually survive to term, and one of them is Down syndrome, which is the triple copy of chromosome 21. And the idea is because chromosome 21 is the smallest chromosome and has the fewest number of genes, so that can be more or less tolerated. So what this group said, okay, we know that X inactivation can silence a whole X chromosome. What if we inserted the X inactivation locus and expressed exist in one of the three chromosome 21 chromosomes in cells that are isolated from a Down syndrome, a person with a Down syndrome, right? Can we actually now silence the whole X chromosome, basically cure the Down syndrome in a dish, right? This is, of course, all kinds of things that you can't really apply to the humans just yet, but this is a proof of principle. So what they did is they took fibroblasts from Down syndrome patients, and they formed induced pluripotent cells that are have trisomy of 21. They used a genome editing technique, which we're going to talk about in the next lecture, where they inserted the exist gene into one of the three chromosome 21s, and then they induced the expression of the exist RNA using an inducible system, and they coded the whole that exist expression coded the whole the chromosome 21 in these cells, and formed this bar body, which is the body that is named that is basically refers to the inactivated X chromosome, and that basically started inactivating the X chromosome. And they looked, okay, now that inactivation, did it actually result in the repression of the genes on chromosome 21? And they found that it was true, that one of the chromosomes were completely repressed. Okay. You can say, so what? You can do it in a dish. What does this have to do with anything? That's true. You might not be able to immediately apply this to the Down syndrome patients, but one of the things that you can do is now you can study the mechanisms that lead to the Down syndrome on a dish, which you can't really do with humans, right? Now you can do experiments, and you can have isogenic control versus patient cells, and then study what's going on with neurogenesis, what's going on with all different processes, right? So it really creates a very nice tool. All right. So that's humans. We all love humans. We are all interested in ourselves because we're narcissistic, right? But let's pay attention to other organisms. So what happens in flies? In flies, the dosage compensation is accomplished by a dosage compensation complex that is formed by several protein subunits that are shown here, and it also includes a long non-coding RNA that serves as a structural subunit of this complex. And what this whole complex does is in males, it goes and binds and coats the whole single X chromosome in males, right? So this is basically in immunofluorescence analysis to one of these Drosophila dosage compensation complex subunits, and this is the X chromosome in males. And the autosomes are not bound. So one of the subunits of this complex is called MOF. MOF is an enzyme that acetylates H4 lysine 16, that's H4K16 acetylation, and increases the levels of that acetylation on the X chromosome. So this acetylation actually is pretty curious. Where it is that these are two nucleosomes here. The H4 tail actually sticks out from the surface of that hockey puck type of structure, and so when it's acetylated, what it does is it prevents these nucleosomes to come very close and get packed upon each other. Okay, so it prevents the compaction of the nucleosomes. And a very nice demonstration of this was done in vitro, where these researchers took and made a long DNA and deposited nucleosomes on it, right? And then they looked under the microscope, and it looked, and this is a model, it's not the actual structure. It looked very compact. And then they went ahead and acetylated H4 lysine 16, and all of a sudden that structure could not form anymore, and the chromatin was more loose. It was more open. Based on this structural effect, how do you think that H4K16 might help transcription?
Maybe it allows an activator to bind to one of the acetyl groups.
So it could be helping with that. In fact, this acetylation is mostly found in the gene bodies. That means inside the genes during the coding region. Based on that, make another guess what it might help. Because we know transcription factors usually don't bind to the inside the genes, right? Except if there is an enhancer inside an intron. How this might help the transcription? Give me a good hypothesis here. You know that this acetylation opens up chromatin a little bit. I just told you that it's inside the coding region where the transcription is happening. How might it be helping transcription? How might it be helping RNA polymerase? What does RNA polymerase and the elongation factors need to do when the transcription is elongating? Perfect. It needs to remove the nucleosomes ahead. If the structure ahead of the polymerase is more compact, it's going to be harder to remove it, right? And if this acetylation is opening it up so that you can remove it easier, they might be helping elongation. And there's several experiments in the past 3 years or so have shown that that it helps elongation. All right, what about worms? And this is very near and dear to my heart because I work on this. And the dosage compensation complex in worms looks something like this. It's again made up of several subunits. But the core of the complex that is shown here as a 5 subunit core actually belongs to the family of protein complexes we talked about, which are the SMC complexes that contain the cohesin and condensin. And the core of the dosage compensation complex in worms is a condensin complex. So what this condensin complex does is, if you look by immunofluorescence in cells and label the DNA, here it's labeled with blue, what you can see is that it binds and codes to 2 X chromosomes specifically and doesn't bind to the autosomes. Autosomes mean all the other chromosomes other than X. Okay? So it binds specifically to the X chromosome, and what that results in is it results in a 2-fold transcription repression. So we're very interested in how a protein complex can fine-tune the expression of a gene from the full level down to the half level, right? Because it's not an on and off. It's harder to think of a mechanism where you can modulate the RNA polymerase activity precisely 2-fold, right? So what kind of questions do we ask? We ask, how does the DCC specifically bind to the X chromosome? And this is a really interesting question because in all X chromosome dosage compensation, you see the complex only binds to the X chromosome, right? Why is it not going anywhere else in the genome? So this is an important question to ask, because any type of epigenetic regulatory mechanism has to have a strategy for specifically targeting the right gene and not targeting other genes, right? So specific targeting of protein complexes in the genome is a very fundamental question. Then we're also, of course, interested in how the heck does it actually repress transcription. So we do a lot of experiments to see, for example, to understand the specificity of binding. We do chip-seq experiments and look at where the complex binds. So here is an example of one of our data. And what we're looking at is we're looking at the ChIP-seq data from the DAMPI-27 subunit of the dosage compensation complex compared to a control ChIP-seq where we don't use any antibody. What you can see is that there are binding sites along the X chromosome, and some binding sites seem to be more than others, etc. And these binding sites happen to be at the promoters and enhancers in the X chromosome. All right, so I'm just going to show you a model of how we think that this complex specifically targets to the X chromosome. What we found is that there is a short DNA sequence motif, about 12 base pair or so, that is found at several sites on the X chromosome. And it's found at multiple copies in some sites than others. And in very early embryogenesis, these motifs are actually closed. There are nucleosomes that are covering these motifs. And what happens first is that the part of the protein complex containing this protein called SDC2 recognizes these sites. And it binds to them. We don't understand how it does it, but it recognizes it. And once it recognizes these sites, it helps to remove the nucleosomes from these sites. Okay? So what kind of... based on what I just told you, what kind of transcription factor might SDC2 be? It can recognize its motif, probably. That's our working hypothesis. Even when the motif is nucleosomal, it binds to it and opens up the chromatin. Not necessarily. I'm talking about sequence-specific transcription factors that do not care if there is a nucleosome or not. We said that these are really important. Enhancer is a genomic element, right? It's not a transcription factor. Pioneer factor, it's the pioneer. It goes first, right? You can remember that. It doesn't care about the nucleosome. It doesn't care about the horrible conditions, and comes in and needs help from the natives to eat turkey next week, right? But so it opens up the nucleosome, and what that helps is it helps recruit the rest of the complex, which cares about nucleosomes because it doesn't bind to any nucleosomal DNA. And once that recruits the rest of the complex goes to other places that contains this motif. So it's like a secondary spreading that kind of should remind you how exist is spreading, right? These are very different strategies, but at the essence of it is very similar. You basically start at very few sites and then start spreading. And after that, the complex accumulates more and more, and then it spreads to larger chromosomal domains with the mechanisms we actually don't understand whatsoever. So we really would like to know that. And that's it for this lecture, and I'll see you in 2 weeks. Oh, no, on Monday. Yep, not 2 weeks. Come to the class. Monday we have class.